It's lovely to be back at MAP and in London. So you've just heard a wonderful talk on ctDNA. I'll capitalize on the talk. I'll use many of the concepts that you just heard and translate them to colorectal cancer. I'll spend approximately 50% of my talk trying to put in despair that cancer evolution is going to limit our effectiveness, and the other half of the talk trying to give you some hope that exploiting evolution may in fact be applicable, especially using ctDNA. These are my disclosures. So I just said that we study colon cancer. Often I don't need to show this slide, but I think here it might be useful for one specific reason at least, that I'll never stop saying that colorectal cancer can be almost entirely preventable. And this, in this light from what was said before for the other type of disease, here we have tests that we know are effective and have very low misinterpretation rates. For example, colonoscopy or fecal test. So, you know, this is a disease that we study, and I study. I spent most of my life studying in the metastatic setting, but here can be detected and blocked effectively. The other reason I show this slide is that we believe that colon cancer will start off from a single cell with specific genetic alteration in a defined pathway, the Wnt APC beta-catenin signaling pathway, and this will come back many times during my talk. So this is important to remember. Third is that approximately 50% of the patients will present to the hospital with disease that has spread already to other organs, namely the liver. Now, when I started my lab approximately 10 years ago, there was an incredible amount of knowledge on the genetics of colon cancer, but none of that had been returned back to the clinic. In fact, I love colon cancer because a lot of the genetics has been greatly worked out. Now, one achievement that as a whole the community has reached is to place many of these genetic alterations into clinically applicable, at least, markers. Now, whether that translates into effectiveness in therapy, I will not discuss, but this perspective of colon cancer will not change. This is something that as a community we have gained, and all these genetics will stay on and we will build on it. Now, it has been mentioned already, and so I don't need to go into any details, that we can now do genetics of colon cancer using tissue and liquid biopsies. And my group, as many others, have started approximately now 7 or 8 years ago to collect blood samples from patients to study how colon cancer evolved. Now, colon cancer, as I said, used to be considered genetically very well defined, but with very few actionable targets. Well, that's no longer true. It's not a boring disease from this perspective. You can see here and here that several oncogenes have been defined in small population. We heard yesterday a very interesting description of the long tails of oncogenes. And I will, as I said, spend half of my talk describing some advances that were made in targeting a few of those. The first I'll start on is HER2. You very well know HER2 amplification from breast tumors. Well, it was known, as I said before, that HER2 was amplified in colorectal, but whether this would be clinically actionable wasn't clear. And my colleague, and with my contribution a number of years ago, Livio and Andrea at the Institute had discovered that you could act upon HER2 in colorectal. And this led to a clinical trial that was run between Torino and Milano, and was based on the finding in what we call xeno patients that you had to use both trastuzumab and lapatinib to effectively suppress the growth of this HER2 amplified cancer. And then if you do this clinically, you get remarkable responses, as I show here. These are metastatic patients after multiple rounds of previous therapies. You get significant responses, and we reported this very recently. But what I'm interested in showing to you is that we concomitantly start to see rebounds and relapses. And this is not even an update. We will provide an update at ASCO this year. But most of these patients have relapsed, unfortunately. We have a couple of patients that did not, which is very interesting in itself. These were metastatic colorectal cancer that did not relapse after HER2 therapy. But most of them, unfortunately, did relapse. Now, liquid biopsy can be used to tackle this issue. And this is shown here. A couple of patients that we are starting to analyze. The interesting thing is that we make this collection very, very frequently. And this patient, after HER2 therapy in colorectal metastatic setting, developed a PIK3CA mutation that wasn't detectable before. Or this other patient developed a couple of BB3 mutations that were lingering as clones, I think, perhaps in the tumor, and throughout the therapy eventually emerged. We're clearly trying to functionally characterize these mutations and perhaps use them in the future. Now, the second example I want to provide of this part of my talk is what we have done in trying to target TRK in colorectal. Now, translocation in TRK are rare. If you build a large clinical connection, like we have done in Italy, on colorectal, then you will intercept them. And if you do so, you will see that these patients have highly expressed NTRK as a result of the translocation, as we reported last year. And then there are drugs to act upon, and this is called entrectinib. It was provided to this patient, and you probably know what to expect. But what I just want to point out here is profound response and also quick rebound. The interesting thing is, in this case at least, in the first patient that was treated with entrectinib in the colorectal setting, we had a response, so the treatment at least was spot on. And we also discovered why the patient relapsed. So this was within one cycle of one patient cycles of therapy. And we found 2 new mutations in TRK that are responsible for the relapses. These are interesting mutations. They are in the catalytic domain, possibly not unexpected with a kinase inhibitor. And you could imagine that new drugs can be developed to target this mutation. In fact, there is a drug that overcomes this mutation. And the last and final example is the one that I've been, as many of you know about, working on for so many years, which is EGFR blockade in colorectal. This has been used widely. I'm not going to claim this is a particularly effective therapy, but it's taught us a lot of lessons on how colorectal cancer eventually evolved. I'll summarize in 2 slides what we discovered. We discovered and reported a number of years ago that the main mechanism of relapse is the emergence of RAS mutations. And in the second part of my talk, I will use this as an example of how you can exploit clonal evolution in colon cancer. And these account for approximately 70% of the patients. So unfortunately, 70% of the patients develop an allele that currently cannot be easily drugged. And the other 70%, and the other 30% develop domain in the extracellular domain, mutation in the extracellular domain of the EGFR. The first mutation was found by Clara Montagut, then we teamed up. One thing which I love to do in science these days is really to put together big teams. And together with Clara, we found many more mutations, and we are reporting now a follow-up on that study with even more mutations. This is amazing. We carefully, essentially, remap the CTAX to EGFR binding site in patients. Right, so this is one case that I often show, which I think it's relevant for this setting, of a typical treatment with EGFR blockade in colorectal. This is a case that was treated as MASGEN. This is the work of, this is the clinical, the patient was treated by Ryan Corcoran, an oncologist at MassGen. He called me up, said, Alberto, we have this patient under the age of 30. You can see he received quite what would be considered in initial chemotherapy and then several targeted therapy, including EGFR targeted therapy. And he asked us to do liquid biopsy because he had found in one of the lesions at progression a very interesting mechanism of resistance, MAC mutation. Our preclinical work that we did supported then at this point a combination of EGFR plus MAC. They acted upon, the therapy was delivered, and fascinatingly and scaring, this is what was observed. So half of the lesion of this patient regressed, including the lesion that through a tissue biopsy was found to contain the MAC mutation, and half of the lesion approximately progressed. And at that point, there was little to be found by tissue analysis, and so we acted upon with liquid biopsy. You can see the MEC lesion going down in the blood, the MEC clone going down. But unfortunately, and it was not detected because it wasn't in that tissue biopsy, a RAS mutation was emerging and eventually, we think, led to disease progression. So that kind of ends the first half. So, you know, these are, in a way, successful stories. To me, they are incredibly disappointing stories. And what this teaches us is not only how relevant heterogeneity in the primary tumor is, but also, and I think it's possibly more important, and we have seen this also from the previous beautiful talk, how relevant this is when mutations are acquired as a result of exposure to therapy. So I'll spend the next 10 minutes or so suggesting a few ways that we can think about on acting upon clonal evolution. The first is what we call exploiting clonal fitness. And you know, we designed a trial that I'll describe in a second. Then we'll present 2 unpublished studies on the presence of rare subclones and how they impact the survival of patients. And finally, I'll suggest an idea on how we can target the trunk of colorectal tumors. What do I mean by exploiting clonal fitness? I told you that the main mechanism of relapse to EGFR blockade is the emergence of secondary mutation in the RAS signaling pathway. I also told you that these can be picked up in blood. One question that we had in the lab was, when there is a patient that is in this situation, that is, there is the emergence of a RAS mutation, there is clinical progression, we often detect the RAS mutation before clinical progression. Therapy then is either suspended or switched to another chemotherapy regimen, palliative care, etc. What happens to these RAS alleles? And is there something interesting that we can find? Well, there is certainly interesting findings to be made. And one thing we reported is that apparently as soon as you stop the EGFR therapy, the anti-EGFR therapy, you see the disappearance of these clones from the blood. I don't think they disappear. In fact, I think they go below the limit of detection. And interestingly, if you then follow patients that were received multiple cycles of therapy with EGFR blockade, you can see these clones coming back. This suggested to us that there is a clonal fitness regulation that perhaps we can act upon. And based on these findings, we designed what is called Chronos. Now, Chronos is a clinical trial that is based on liquid biopsies, and it is designed in the following fashion. We will track the rise of RAS mutation in the blood of the patients. When there is clinical progression, we will continue to closely monitor. And then when, and this has been arbitrarily set, when there is a drop by 50% in the initial level, the maximum level, we will restart anti-EGFR therapy. Now, do I think that this will fundamentally change the clinical outcome? I don't, but I think we will learn a lot, at least on how you could potentially re-challenge with the same therapy. We'll see how it goes. The trial has been funded and it started. The second idea on how you can exploit, perhaps, evolution is the following. You can use it to predict survival, perhaps. Now, the initial ideas on this came us from liquid biopsy. I told you that there is 2 mechanisms, RAS mutation and EGFR Ecd mutation, and that these events sometimes co-occur in patients, not surprisingly. They may occur in different lesions. But what we started to see is that often we would, in patients that develop both of them, we would often see first EGFR Ecd mutation rise and then RAS mutation. Why was that? We were intrigued by these findings, and we follow up. Again, we had to team up with a big number of clinicians, and so we analyzed, in this case, we had tissue pre and post, and what we found is that in the patient that developed RAS mutation, if we went back to the tissue, we often could see it in the pre-treatment samples. And fascinatingly, we found a correlation between the relapse time, so the progression-free survival, and the presence of either RAS or EGFR Ecd mutation prior to therapy. In fact, what we found is that almost never, as I will describe in a second, also, almost never we detect EGFR ECD mutation. In fact, so far never, but you cannot say in medicine never, but we have yet to find it. And certainly we confirm what was suggested by liquid biopsy. That is, patient will have a different PFS according to which mutation they develop. So we went back to the lab and asked how we can model these in cells. So we took cells that were sensitive to EGFR blockade, labeled them with genetic tracking barcodes, and let them go. And what we found is that depending on the selective pressure that you apply to these cells, RAS mutation can rise in the absence of EGFR blockade with an antibody. For example, if you simply starve the cells or reduce their growth access to their access to growth factors, they will spontaneously develop RAS mutation. However, they never spontaneously develop EGFR ECD mutation. Actually, that's probably wrong genetically speaking, but we don't see this mutation in the cells. And so Beth Vandenberg, she's the leading author of this work, went on to show that actually we can model this in preclinical setting and see the rise of these alleles differently in the population. And so a model that we suggest is the following. I no longer believe that there is a KRAS wild-type colorectal tumor. I think, unfortunately, most of colorectal cancer carry low level of RAS subclones. The reason they exist, it's unclear, but it could be that in the ecosystem, they just are not fit to exist. On the other hand, the EGFR ECD mutation do not provide any advantage, apparently, in this setting. And they, of course, instead provide a big advantage when you apply EGFR blockade, and therefore they rise later. Of course, this is hypothetical. I'd love to go deeper into these findings because I think they are fascinating. I'll move on the third. How much time do I still have, Charlie?
Well, I'd like to listen to you forever, so just carry on.
All right, all right. 10 minutes, so I'll be done in 10 minutes. So the other suggestion that we have is perhaps you can act upon clonal evolution, okay? And one way to do this is to use detection in the blood to direct therapies. Something that is fascinating, right? So we all talk about liquid biopsy, but the truth is, and I think Nick just showed it beautifully before, you need to design a trial in which you act upon a liquid biopsy to direct therapy. This will be a way that you can prove that they are really effective. So we thought this could be an interesting setting from that perspective. I told you that we discovered this mutation, and I told you that we can detect them in the blood. Now, the interesting thing about this mutation is that we know exactly where they map. In fact, there are many more than the one that I show you, because we continuously discover one mutation in the same amino acid or close by. EGFR and panitumumab will bind, obviously, right there, but there are other drugs that bind into different position of the EGFR extracellular domain. And many of these drugs, which are typically oligoclonal drugs containing multiple antibodies, will be effective against this mutation. We also have shown, taking a xenoph from, deriving a xenoph from a patient that developed this mutation, that even if cetuximab and panitumumab are ineffective, these drugs are clearly effective. So now the idea is, well, occasionally we will find a patient in which we apparently don't see RAS, but we do see EGFR ECD mutation. And so perhaps when we find this mutation rising in the blood, we can act upon. Now, to prove, at least conceptually, that this would work, we retrospectively analyzed a large collection of serum. In fact, it was serum, not plasma, but liquid biopsy can be done in serum occasionally, of patients that, for other reasons, were treated with these drugs. Some of these patients had responded, and there was no correlation between the treatment and the response. So one fascinating finding we had is that in 3 at least of these patients, we went back and found that when they responded to the MM151, they had previous evidence of EGFR extracellular domain mutation. Again, do I think that clinically this will fundamentally change the history of these patients? No, but I think it will be still very important. And so this led to what we call the EGFR ECD trial. It's sponsored by the company that make not this drug, but a very similar drug, which is called SIM004, and also has been funded. And so, you know, plasma will come to us. We will have 15 days to act upon. This is a big challenge for my lab. We decided to take it because it was the time to do it. So for the first time, together with the pathologist, we'll have 15 days to report back to the oncologist the findings. And if there is EGFR mutation and no RAS mutation, they will go on and treat. Finally, in the last 2 minutes, this is really more conceptual. We don't have that many data, but I think it's important to discuss is the idea of targeting the trunk. All I show you up till now is that, you know, targeted therapies can be very effective in reducing the bulk, but unfortunately, eventually, it comes back. And when it comes back, as Charlie always reminds us, often it is even more aggressive and more complicated, right? So a question that I have, and you know, which I think is important conceptually, is does a cell that has developed several resistant mechanisms, I show you how many mechanisms a cell can develop, remain addicted to the initial event that made it a colorectal tumor? In this case, this event is an APC mutation. Now, unfortunately, as we all know, there is no easy way right now to act upon an APC mutation, but fascinatingly, the pathway is much more complicated. And this is a little bit of a school animation, but that shows you how it works. So essentially, there is a fraction of colorectal cancer in which activation of the APC Wnt pathway doesn't occur by truncating mutation in APC, but by alteration in the Wnt signaling pathway upstream. And these tumors do not have mutation in APC. So the two things are mutually exclusive. The other thing we know is that there are drugs that have been developed to target this alteration. For example, R-sponding translocation. And so we ask a very simple question. Can we find cells that would have this alteration? Can we make them resistant to a targeted therapy? Would they remain sensitive to Wnt signaling blockade? So we went back. We now have a collection of about 250 CRC cell lines and found a few. There are very few, but there are some that are sensitive to these therapies. So that are sensitive to two therapies. So we had to find a cell that had a druggable oncogene. In this case, we found a BRAF mutation. And also that was sensitive to Wnt pathway blockade. So these cells were hard to find, but we did find a few. And so you then start with the cell that is sensitive to the targeted therapy, in this case, BRAF inhibition. You make it resistant to BRAF blockade. These cells will develop RAS mutation or BRAF amplification. It doesn't matter here. They develop multiple mechanisms. And then we ask the question, will they be sensitive to the trunk, to the initial event? And you know, the data that we have so far are conductive. I mean, they do remain sensitive. Now, you may say, well, this is obvious. Actually, it wasn't from my perspective because the pathway often crosstalk. And so, you know, this could be an interesting way of some sort of, you know, evolutionary synthetic lethality that is being created. With that, I will close, and I want to thank the people that did this. Sandra, essentially, as a PhD student, discovered the RAS story. Mariangela did the NTRK story. Bat, I mentioned for the clonal evolution and fitness. These are all, Julia Siravegna, she's a brilliant PhD student, did the liquid biopsy work. And these are the collaborators at the Institute and outside. Thank you very much.
Alberto, that was brilliant, as always. Given Nick's talk, data on the prevalence of ESR1 mutations based on the timing of start of aromatase inhibitors, and I apologize if you've published this, but do you have data on the prevalence or the mechanisms of resistance specifically to EGFR blockade based on the timing and the sequence of treatment?
No, we don't. But it's incredibly important to gather this data. And now that we know the mechanism, we can use some of the collection that we already have to see how they rise. But personally, we don't.